Okay, so good afternoon, everyone. It's a pleasure to be back here to this great meeting, and thanks Francesca and team for the opportunity to speak here today. Today, I will tell you something a little bit different that we have heard so far, and particularly I talk about the PCDH19 function in the non-cluster pro-adhering combinatorial code. And also, we will mention at the end of my presentation our working progress involving a conditional mouse model of PCDH19 and how we can use this model for silencing PCDH19 in the regions of the mouse brain and also in the cell types. So, as my colleagues already introduced the topic very nicely, I'm not going to spend much time on the background, but I really want to mention some aspects that will be relative to the rest of my presentation. So particularly PCDH19 gene, as you know, it's a large gene that consists in 6 exons, and the exon 1 is the largest one, and the majority part of the mutations that cause the disease phenotype are actually found clustering in the exon 1. And as I said, the exon 1 encodes for the transmembrane, the extracellular domain of the protein. And our laboratory, we are particularly focusing on this extracellular domain and the function that it plays in the cell-cell adhesion properties. So as a calcium-dependent molecule, PCDH19 belongs to non-cluster subfamily, which in turn is divided in 3, depending on this the homology that the members present in the family, together with the number of extracellular adhering domains. And what's been shown that actually these non-cluster pro-adherents, they are not expressing the separated cells, but through the in situ hybridization has been seen that there is overlapping spatio-temporal expression of all these non-cluster pro-adherents during mouse development in the brain. And that actually brings the idea of the combinatorial expression of PCDH19 and non-cluster pro-adherents, and that potentially plays an important role in this brain architecture and physiology. So how is actually non-cluster pro-adherents interact together, how they promote these adhesion properties? So probably many of you are familiar with this model, which is called the zippering model. And based on that, there's a 2 type of interaction. So there's a cis interaction, which occurs between the extracellular adherent repeat 5 and 6 between different non-cluster pro-adherents. And actually, this is a very heterophilic interaction because it occurs between different non-cluster pro-adherents. However, the interaction that actually drives, okay? So I said there is an overlapping expression of non-cluster pro-adherents, which have been found in the mouse and rat brains, just proposing that there is a combinatorial expression of these non-cluster pro-adherents during development with the potential role in the brain architecture and physiology. So again, I already explained pretty much the model that's been proposed for a cluster pro-adherence, but apparently the non-cluster pro-adherence interacts in a pretty similar way. So I'm going to go back to this trans interaction that I said as interaction that occurs between 2 cells in the antiparallel orientation. So we have this happening between the extracellular adherent repeats from 1 to 4 from 2 different cells. And it's been shown that actually this interaction, or proposed that this interaction is strictly homophilic. And I will, later on my presentation, I will show that that's not always the case. There are some exceptions. And on the right, as you can see, there's some example that's aggregation assay that's been performed in our laboratory by Daniel Pedh, who actually shows the adhesion abilities of different non-cluster pro-adherents, including PCDH19, 17, and 10, showing that is when we express PCDH19 as a single molecule with the 2 different under 2 different reporters, a mixed cell population that express each of these molecules and the different reporters mixed together, we found this heterophilic cluster formation, which indicates that they form the proper cell-cell interaction. But when we have cells that express only one non-cluster pro-adherence, for instance, 19 is last example in here, And we mix the cell population that express these cells with another population of cells that express PCDH10, so a different non-cluster pro-adherence. You can see that the cells segregate apart. So there's no adhesion adhesion code between them to interact. But that's actually just in a single cell and as a single molecule. And in our laboratory, we currently are more interested to understand what's the role of PCDH19 in the non-cluster pro-adhering combinatorial adhesion code. So to answer this question, we employed a technique called probabilistic cell typing by cytosequencing. I thought somebody from the IT is calling over. And that technique actually allows us to identify up to 100 genes in the same cell. And you can actually trace these cells and see what's their localization in the brain, because the tissue morphology is conserved. And this was performed in Karolinska Institute. So we used two sets of props. One set of props that identify all the members of the non-cluster pro-adherence, and another set of probes that identified the different neurons, so excitatory or the inhibitory neurons. We used about 15 different markers, but I'm going to show you an example just for the most relevant of them. So the first more global analysis of the data analysis is summarized here in this heat map that shows that in the hippocampus, PCDH19 it's co-expressed with the PCDH1 and 9 in a high number of cells. And in the cortex, PCDH19 it's highly co-expressed with the PCDH9 only. So next, we wanted to know which cell type actually expressed PCDH19, and that's the first filter that we applied. And we found that actually glutamatergic neurons in the cortex and hippocampus express PCDH19, and that's the most dominant cell type. We also have seen that PCDH19 is expressed in the somatostatin positive neurons, but we haven't seen any power bombing cells that express PCDH19 in cortex and hippocampus. So next we perform a little bit more specific analysis. We wanted to know which two non-cluster pro-adherents are expressed together in the glutamatergic neurons. And this is the heat map of the summary of this data in cortex and hippocampus. Probably we will ask a question, why just two? Because they could be 3, 4 more. Because the technique that we used allow us to test, allow us to identify up to 5 members that's been expressed in the same cell. However, in the cortex and hippocampus, we found that the majority of the cells expressed only 2 non-cluster pro-adherents. And when it comes to the 3 members of the family, we found just individual cells expressing that number of non-cluster pro-adherents, and almost no cells being found to express 4 or more non-cluster pro-adherents. And that's the main reason why we restricted to this analysis just to 2 non-cluster pro-adherents. Again, here we are particularly focusing our research on PCDH19. So we wanted to know which glutamatergic excitatory neurons actually co-express PCDH19 together with any other non-cluster pro-adherents in the cortex and hippocampus. And we found that in the cortex, PCDH19 is expressed in a high number of cells together with PCDH9. And in the hippocampus, we can see that PCDH19 is co-expressed in a high number of cells together with 1 and 9, but also PCDH17 and 18 are co-expressed with the PCDH19. For the next part of my presentation, I will be focusing mainly on the interaction between PCDH1 and 9, and PCDH19 and 1, and PCDH19 and 9, because in our lab we previously have shown this interaction between 19 and 17 and 19 and 18. So firstly, we wanted to evaluate what's actually the role of PCDH19 in the combinatorial code. It's formed by PCDH19+1 and PCDH19+9. To do this, we again perform this aggregation assay that I have explained to you before. So that consists in mixing 2 cell population, expressing firstly the same non-clustered pro-adherence, so in this case PCDH19, PCDH9, and PCDH1, and they're expressed under different reporters, so we can visualize the intermixing abilities. And as you can see, when the cells express the same non-clustered pro-adherence, they mix together. However, when we mix the cell population that express only PCDH19 with the cell that express PCDH1 only or PCDH9 only, this cell population segregate apart. So we have these homogeneous clusters of each of these populations, so they cannot intermix because of their not compatibility as a single molecule. However, we performed the immunoprecipitation assay, and actually this assay being performed in Joseph Get's lab by Raman Kumar, who found that actually PCDH19 physically interacts with PCDH1 and PCDH9. And that's the first time with actually being shown this interaction with a member of delta 2 non-clustered pro-adherence with 6 extracellular adherent domains interacting with the members of delta 1 non-clustered pro-adherence subfamily with 7 extracellular adherent domains. So it's a little bit interesting in that perspective. So next we evaluated what's the role of PCDH19 in this combinatorial expression, and to this end we perform a mismatch aggregation assay. So it's pretty bit similar principle. So we have the cell that express the complete combinatorial code, that's 19+1, that's expressing with M cherry, and the cells expressing the same code with the GFP. And we found these cells mixed together, so again through these heterogeneous clusters. And here is the quantification, and that is our indication for 100% adhesion abilities. When we have the situation when we mix a cell population expressing the complete code with a cell that expresses only PCDH1, we again see that actually this adhesion ability almost remains intact, so it's insignificant compared to the control. But when we mix cells that express the PCDH19+1, so the complete code, with a cell that express only PCDH19, these cells segregate apart, which means that this combinatorial code actually present a different sensitivity to a specific molecule that is expressing the code. Next, we perform a very similar assay, so in this case to evaluate what's the role of PCDH19 in the PCDH19+9 combinatorial code. And again, what we found here in a little bit different scenario. So when we mix cells that express the complete code that you can have here, with the cells that express PCDH9, that means 19 is missing, because I have forgotten to mention at the beginning that the cell line that's been used for this assay doesn't express any adherence, so whatever it's introduced endogenous, that's it. In this particular situation, when we have cells expressing the complete code with a cell expressing PCDH9, we have missing PCDH19, and that's actually a scenario that we have observed in the brain of a heterozygous PCDH19 mouse model, probably in humans as well. We still don't know exactly. We can see here that actually PCDH19 in this specific combinatorial code plays a dominant role, because missing it causes cell segregation apart, so to different clusters of cells that can be seen in the dish. On the other side, when we have the cells that express the complete code mixed with the cells that express only PCDH19, you can see that actually this combinatorial adhesion interaction between the cells expressing the complete code and the one with the 19 remains intact. And actually, this is very interesting point here, because what we found that beside of the similar cis interaction that's been found with the PCDH19+1 and PCDH19+9, we still see some differences in terms of trans interaction. So PCDH19 actually plays a very different role in the cell-cell interaction in the two different codes. So in the 19+9, it's a dominant one, who's sufficient to maintain the interaction between two different cell populations. However, in this scenario, 19+1, there's a different, in that case, PCDH1, who is actually driving these adhesion abilities. The another interesting finding in this experiment using the cell, this in situ sequencing, was to find that actually cadherin 13, who is an unusual member of the cadherin family. Unusual, it means that it doesn't possess any transmembrane or intracellular domain. It's been found to be co-expressed with almost all the non-cluster procadherins. And particularly, we found that in cortex, there are moderate number of cells that co-express in PCDH19 and cadherin 13. And again, when we applied just to see which cell type are actually expressing this cadherin 13 plus PCDH19, and we found again that they are mainly glutamatergic neurons. We found it so interesting in terms of the disease, because it's been, we found a publication that's been published in 2019, when it's been described that actually PCDH19 as PCDH9 and cadherin 13 are involved in the seizure and epilepsy. So it's actually there a link, could be potential link to the PCDH19 clustering epilepsy. So again, we evaluated what could be the role of PCDH19 in this combinatorial adhesion code. So firstly, what is important to mention here is that cadherin 13 presents a very weak adhesion abilities when it's by itself. However, what we found that when we express the cells that, when we mix the cells that express the complete code, so we have PCDH19 and cadherin 13 in the same cell, that actually promotes, turns this weak activity that's been seen in the cadherin 13 in the pretty strong adhesion abilities between the different cell population. And we believe that actually this is because of the cis interaction that's been already demonstrated between PCDH19 and cadherin 13. So next we perform again this mismatch aggregation assay to see what's the role of PCDH19 in this combinatorial code. And again, we've seen when we mix cells that express the complete code with the cell that express cadherin 13 only, which means that here we have a missing PCDH19, let's say more artificial PCDH19 clustering epilepsy scenario, and we see that the cell segregate apart. So the adhesion abilities to interact the cells that express only cadherin 13 with the cells that express completely the complete code, it's affected. However, when you mix cells that express the complete code with the cells that express only PCDH19, again, this adhesion ability is restored. And actually, that means that PCDH19, again, plays a very different role in a specific combinatorial code. So next we wanted to investigate whether actually PCDH19 as a dominant protein of composing two different heterophilic codes, it's enough to drive the interaction between them. So we mix cells that express PCDH19 and cadherin 13 with the cells that express PCDH19 and PCDH9. So in the both codes, PCDH19 plays a dominant role. And we have seen that actually that is enough to maintain the adhesion abilities between them. That's something that hasn't been demonstrated so far. On the other side, when we mix, and that's our control, when we mix cells that express PCDH19 and cadherin 13 with the cells that express PCDH19 and 1, again, we have a different scenario. So in this particular combinatorial code, PCDH19 is the dominant one, but with 19 plus 1 is the dominant one. And you can see this segregation of the cells. So there is not interaction occurring between two different heterophilic expression. We have seen the pretty similar outcome when we mix cells that express 19 plus 9, when 19 is the dominant one, with a 19 plus 1. And actually, we found this very, very intriguing because it's showing that actually this heterophilic interaction, it does exist, but depends on which dominant protein it's actually expressed in each combinatorial code could drive this interaction or could just disturb it. So to sum up our data, what we found is that PCDH19 is being found co-expressed with PCDH9 and 1, and also with the cadherin 13. We found that PCDH19 is a dominant protein within the 19 plus 9 and 19 plus cadherin 13 combinatorial codes. We also found that actually, as a dominant protein, PCDH19 could drive the heterophilic interaction between two different combinatorial codes. There is also a finding that PCDH19 interacts in cis with the PCDH19 or CDH13, and we believe that actually this cis interaction between 19 and 13 it's important to promote the CDH13 adhesion abilities. So I'm going to show you some very briefly our work in progress, and that has started about a year ago. So for those of you who don't remember, like 3 years ago, we have established a conditional mouse model, so pretty similar that Sarah showed us like a couple of hours ago, but in this case we have flux exon 1 of the PCDH19 gene, and we have a GFP reporter who's been placed after the 3' LOXP site. So after the pre-mediated recombinase, we see that the expression of GFP and elimination of PCDH19. So we also tested whether this genetic modification could affect the endogenous level of PCDH19 on mRNA level as well. We haven't seen any differences, which means that it's actually not making any changes in the endogenous expression. So firstly, we tried this model to silence PCDH19 in all relevant neuronal cells by using SYN1-CRE line. So here is like the outcome, what we can see in 3 weeks after birth. So it's pretty stable GFP expression, which actually demonstrate knocking out PCDH19 in the most relevant part of the brain. So we investigated it is pretty similar that we have seen in the previously established PCDH19 knockout mouse model. And we found that actually silencing PCDH19 in the neuronal cells accelerate the neurogenesis. We tested 2 different markers, K67 and double curtain, and in both cases we see increase in the neurogenesis in this double head mice. Currently, we're investigating how that could impact the behavior, the mouse behavior, and also we are planning to perform some electroencephalograms to see whether there's some changes in the brain activity. That we'll be performing in collaboration with Steve Petro in Melbourne, not in our laboratory, but that's still work in progress. Hopefully next time we're gonna show you more data related to that. Yeah. So I'm gonna jump to the next part, which is like region-specific deletion of PCDH19, which is also quite interesting because what we found in the literature is that this general astrocyte marker GFAP, as you probably know, during early neurogenesis, is highly expressed in the neuronal stem cells that gives rise to hippocampal astrocyte and neurons as well. So we decided to use this mouse line to silence PCDH19 in the hippocampus, first to see how that works. And as you can see here, after breeding PCDH19 flocks with GFAP CRE mouse line, there is a restricted GFP expression of into the hippocampal area. And we found that many of these GFP positive cells are neuron positive, and we also have seen some GFAP, some astrocytes that are also GFP positive in the CA1 area that I have previously found as area that there's some astrocyte also expressing PCDH19. And again, we evaluated the neurogenesis, what really happens in this particular mouse line. And we found again that actually silencing PCDH19 in the hippocampal only area, that's also accelerate the neurogenesis. And next we wanted to know whether that somehow impacts the learning and memory formation, because we know that the hippocampal in the hippocampal area, we have the PCDH19 knockout cells, but this area interacts with the cortex through the entorhinal cortex. So that area, we're going to have PCDH19 wild type cells. From our previous study, we have shown that actually the interaction between the knockout, PCDH19 knockout and the wild type cells actually alters this neuronal communication. Not just us, but as you can see, our colleagues also confirming the same discovery. So we perform a pretty much very simple, I would say, behavioral test, which consists, it's called a novel object recognition test, and actually helps to assess this learning and memory formation. And the test consisted putting the mouse in a cage when we have two identical objects, and leaving the mouse to get familiar with the objects, play with them, etc. And 24 hours later, we put the same mouse in the box, but in this case, we have the familiar object plus a novel object. And it's the rodents usually tend to explore the novel object more than the familiar object. It does the way that we can assess whether the learning formation is affected or not. And what we have found here is actually that when it comes to the exploring the novel object, we haven't seen much differences between the two strains. But when it comes to the exploring the familiar object, when it's, as you can see here, the control animals actually been able to recognize that is their familiar object, and they spend less time interacting or playing with this object compared to with the novel object. However, these differences hasn't been seen in our experimental mouse model, which actually suggests that this the memory formation in this particular mouse strain is affected. So silencing PCDH19 in the hippocampus could affect the learning and memory formation. So again, we want to perform more behavioral tests just to be sure which parts are actually more affected. And again, with the electroencephalogram, we want to assess whether there are some changes in the brain activity. So I'm going to stop here. I would like to acknowledge the members of our team, led by Professor Paul Thomas, particularly Michaela Skerer, who assisted with the sample collection for this study. Also, our long-term collaborator, Professor Joseph Getz and Raman Kumar, who performed the IPs. Our collaborators from Karolinska Institute and our funding bodies. So thank you all for the attention, and we'll be happy to have any questions.
Beautiful data. That's good. So with that last part, with the novel object recognition, I mean, they still spent more time exploring the novel object overall. So are they just hyper-exploratory, or what does it mean?
They what they actually do is spend the same time, pretty similar. So here we have with the novel object, they spend like 30 seconds. With the old object, it's around 20 something. So pretty similar time. But of course, that's the n number here plays an important role, because that's a pretty much preliminary data. And as you can see here, we have just 4 samples, well, so 4 mice that's been tested. Usually for the behavioral test, just to make the differences smaller or whatever, we need a higher number. So I don't think there's a big difference when it comes to that exploring more time compared to the controls. I just believe that probably that will be decreasing with increasing the number of animals that are involved in, because usually it's considered around 20, any time between 20 and 40 seconds for a normal and average time of playing with the object. So I think they're between the normal range. So I wouldn't be saying that they are really playing too much with the novel object. But it's like most importantly what they are doing with the familiar object, whether they are playing or spending the same time as a novel object, whether they can recognize that there is something that they have seen before, or they just ignoring it and treated it as something new.
Question. So when you say that, so I assume you're basing your statement that the interaction exists based on the COMP experiment, which is overexpression of 2 protocodyleins in from 23 cells and then
yes.
Are you assuming that they're interacting in cis based on the results on the cell aggregation experiments? Because in principle, once you lyse the cells, the molecules are floating around.
Yeah.
So they could be interacting in many different types of ways, not necessarily. So I'm not sure you can say it's the two molecules on the same cell that are interacting together.
You mean, but is if the interaction occurs before, that you'll be still, if they're interacting together at the beginning before the lysis?
Well, but yes, but they could be interacting. I mean, you have a lot of cells in the dish.
Mm-hmm.
Usually, two nucleuses are going to be pretty
dense.
confluent. Yes. Dense.
Yeah, so this specific question, I think that because of the eyepiece that's been performed with Raman Cooper, maybe he can answer more properly the question. But what we have seen, specifically with PCDH19 and CDH13, we have seen is just, but that's not for, doesn't demonstrate that cis interaction. That is co-localization of PCDH19 and CDH13, but it doesn't say that actually this is a cis interaction. But I'm not familiar with any other way of showing it in terms of cis interaction if it's not an IP. So just
no,
more.
What I'm saying is I'm not sure whether an IP can 100% tell you whether an interaction is happening in cis. If you have cytoplasmic proteins,
yeah.
Probably yes. But
Yeah, you mean the fractions, yeah.
Yes, yeah.
Okay, yeah, it's not.
I mean, for trans, you could transfect proteins labeled with different epitopes on different cell populations and put them together and then lyse. For cis, I think it's a bit more complicated.
Okay.
These protein, these genes are still not detectable from single-cell RNA sequencing studies because those co-expression experiments could be kind of validated on the single-cell RNA sequencing, but that's still probably not possible. Or it's still below because I remember we were discussing it at some point that all the single-cell RNA sequencing
needs to go too deep
not to see them.
Yeah. That was the reason why we chose this technique, because of the identification capability of the non-cluster proadherins, because we discussed with other researchers, not exactly for non-cluster proadherin, but in general, and they actually suggested that it's much better because of the RNA-seq specifically for a non-cluster proadherin, seems that it doesn't work very well in terms of needs to go too deep to read it, yeah.
But the data from the Allen-Brain Atlas, they do show expression, at least for protein 19, that you can check, and there is actually expression on that data set in parvalbumin intermediate, whereas you said that you didn't find it?
No, we haven't looked this way. It's just a discussion that we had.
At the very beginning, when you were showing the results, you said that you could find protein 19 expression in somatostatin positive intermediates, but not in parvalbumin positive intermediates.
Yes, exactly what I said.
But the data from the Allen-Brain Atlas show expression of protein 19 in parvalbumin expression.
Yeah, I've seen that. It's just was wondering whether this expression is like happening at the embryonic stage, what we are actually investigating. So it's 18 and a half, or it's later, I don't know.
So you were looking at 18 and a half.
18 and a half.
Allen-Brain Atlas is adult, I think.
I also believe that it's like the postnatal stages. I'm not sure about the embryonic stages, whether there is a expression or something.
Yeah, there's not much parvalbumin expressed in that early.
No.
Yeah.
Because the endogenous level will
have to go to P10, P12, even P20 to see that.
maybe
so
later.
There might be co-expression.
Yeah, maybe later on. Yeah. It's just was, I had to specify that we are particularly focusing on this stage, this embryonic stage. We are not investigating anything later on.
But is PCDH19 deletion in hippocampus mice or mouse? Did they also have seizures in either?
No, we haven't seen.
No,
we haven't seen any spontaneous seizures. It should be inducible because I don't know any research that could show that hetero heterogeneous expression or the mosaic expression of PCDH19 actually causes spontaneous seizure by itself. It's like without to be induced. So
Okay.
no,
So
neither
have
this
you
one.
done any EEG syndrome?
No, they haven't tried yet. But
No
yeah,
EEG recording on the.
not yet, not yet. That's work in progress. We just started.
Yeah, do you think the increased neurogenesis is activity dependent? Have you looked at CFOS or anything?
No, I haven't. See fast. No.
Could be these things.
Yeah, exactly. That's a good idea to assess this. We also don't know whether that's the neurogenesis, whatever, or how it's affected, whether it's randomly happening, and whether disturbing any electrical activity. So what's the impact actually in the physiological level? We haven't investigated it
Yeah,
yet.
6,000 things increased neurogenesis.
It's increased or premature?
Oh,
I mean, it's early or it's generally increased, but it's not earlier.
we measured it at P21 for the SIN1 CRE and P42, so it's a little bit late.